I won't, as I'm presenting, I won't really be able to see the chat, but I'll look at it at the end. And please stay on mute for the presentation. And at some point, if my internet gets a little funky, I might ask everyone to just turn their videos off. Um, but yeah, we'll see how it goes. And I guess we'll get started. So, my name is Kunal, and my dissertation is titled Soil Monologues: Critical Ecotones of Pollution, Health, and Justice. As you listen to my lecture, I want you to reflect on your relationship to land, to soil, health, and in particular, the intersecting layers of oppression that create pollution and the overlapping solidarities that disrupt pollution. Take a breath. I'm glad that you're all here. And this is transdisciplinary work, so I'm going to be translate as best I can to all the different audiences. Um, but there's a lot I'm going to go through. This is essentially like four lectures smashed into one. So I hope at least one of the sections resonates with you. And let's go. I want to start out by reading my dedication. So I dedicate this research and this work to the people and lands who made it possible, to all the community members, promotoras, students, kids, families, and teachers who are the core of this research. I dedicate this work to all the people and places that take a stand against police terror, genocide, land theft, resource extraction, and supremacy in all its forms. May we work towards a liberated world that we envision, free of oppression and free of pollution. For this talk, I'll go into my positionality or how I enter and approach this work, and I'll give some background, and then we'll get into what the research was and what it says. So, if anyone knows me, they know I'm a very reflective and often deeply critical person. And that shows up in how I approach science, how I approach my identity as the work. There's so much I could say, but I'll just briefly name that I care about the land, I care about liberation, whether that's of us from carceral systems, whether that's us from the control of corporations. I believe in it all. Some of the fields that really guide my work are here, shown in this Venn diagram: critical environmental justice, community science, political education, and anti-colonial pollution science. And at the core of all of this is an understanding of multiple truths. There's a Jain word that encapsulates this called anikantavara, and it means that there's no one way to see the world. There's no one correct truth, and we exist at the many intersections of how we see the world. And to that end, there are a lot of intersections and overlap between the work that I do on resource extraction and pollution here to liberation movements around the world and in Palestine. This dissertation is focused mostly on mining pollution in Arizona, and some of the very minerals mined here go towards making weapons that are sold and used against Palestinians. And so I hope in that a small part of this work can contribute towards pushing back against the colonial and capitalist systems that cause and result in harm and oppression and colonization around the world. And in that vein, research shows that colonization and settler sciences result in extractive land relations. This graph by Dr. David Chavez shows the interaction between forces of indigenous regeneration and oppressive forces. And we can, in fact, lean more towards the forces of indigenous regeneration. But the colonial and settler worldviews and ways of knowing, rooted in narrow, highly empirical, extractive, and capitalist and carceral values, are mass produced at a global scale that then cause extraction, exploitation, consumerism, and more. All of this leads to widespread environmental pollution that affects every single aspect of our lives, including our soil and including our food. In Arizona alone, there are over 400 mines and mining sites contributing pollution to air, land, soil, and water. Um, some of the major minerals that are mined in communities we work with are copper and gold. These are some photos of these mines. And the impacts of mining don't end once the mining itself is over. The resource extraction leaves long legacies or afterlives of pollution that continue to harm people on the planet, not just physically, but also culturally and politically. But in this very context, there are also forces of regeneration that are existing to push back against the oppressive ones. Things like cooperation, sacredness, land back, and even labor organizing are ways to push back against resource extraction and the colonial systems. A revolutionary approach to our science, to our work, is what can get us out of an extractive system. And so this research is community and citizen science. We're working with environmental justice communities across the country, mostly in Arizona, to show how pollution poses a barrier to community health. And we strive to address these health injustices in a community-based, mixed methods, transdisciplinary way that centers the needs and the futures of environmental justice communities. We're not just studying the past or past harm. We're trying to figure out how we can move forward into a future that is less harmful. So in this context, people are trying to cultivate right relationships with land, preserving traditional growing methodologies like harvesting rainwater, growing cultural crops, and again, trying to build a relationship with land. Um, and using a community science framework, working with promotoras or community health workers, where community members are involved in nearly every step of the research process, from defining research questions, figuring out methods, collecting soil and plant samples, as shown here. We hope that this work can be a part of empowering communities uh, to fight back. And I'm happy to talk more about these methods if people have questions later on specifically, but I'll leave it there. But once samples are collected by community members, they come to the lab, they're analyzed for metals by me. And here's me washing some plant samples, getting ready to dry and analyze them. But as I said, this work isn't just about understanding the harm that has happened already, but understanding what we can do about it in the future. And so another huge part of our work is to lead youth trainings and capacity building in the communities we work with, using frameworks of environmental health literacy and a social ecological model of health. And last summer in June 2024, as part of a whole host of different activities, we held two trainings with young people in Hayden-Winkelman and in the Globe Miami areas. And these are photos of us at those trainings. And we covered so many topics, from environmental justice to environmental monitoring. We were doing Do It Together, sample analysis with soils and water that students collected. But we also had a lot of deep conversations about what environmental justice means, what culture means, and what it means to act. Students also did data visualization, presented their work, photo voice, and so much more. Um, and, okay, I just shared a lot of methods and a lot of the background of what this work is, and it has resulted in an enormous amount of data. From 2010 to 2020, community members collected over 577 rooftop harvested rainwater samples, which isn't the focus of this presentation, but it was still part of my dissertation, so I wanted to highlight that. Over 435 soil samples and almost 1,000 garden plant samples. And then in 2024, 49 young people contributed to 36 matched pre and post surveys, focus groups, interviews, photo voice, workshops and posters, advisory boards, and much, much more. And I want to highlight that all of this and what I'll be talking about today is like a fraction of the amazing work that happens in the lab and is a part of a constellation of efforts that include interactive websites, newsletters, art installations, workshops, presentations, trainings, extension articles, science nights, the advisory boards, reports, talks to school boards and town councils, phone calls, and so much, so much more. Um, and yeah, this is just a fraction of what we do. So with all of that intro, let's get into the results of it all. So as I said, this work is going to focus on the soil and plant samples, and then I'll focus in on all the data from the young people. So the first objective of the dissertation was to understand the major drivers of rainwater, soil, and plant pollution in marginalized communities, mostly in Arizona. And the research shows that community is by far the most significant variable. In all the models we ran, probably over 1,000 different models. Season then came in as the next most significant variable generally, and then point source of pollution, and then various biogeochemical properties like pH, and plant morphology. But I want to emphasize that community is a stand-in for a bunch of other variables, things like climate, geology, pollution source, and uh, even the types of plants grown in the garden, it all varies by communities. So when we see community pop up again and again and again, it really emphasizes the need for local community-based solutions and frameworks to address these issues because things are so different in each town you go to. And these broad generalizations that settler colonial science loves can't actually help people at a local level. The second objective was to determine the specific health risks, human health risks associated with growing and eating crops in contaminated environments. So we modeled a high risk situation where someone might be eating a salad of 20 different, of the 20 most commonly submitted plants in our 19 most commonly submitted plants in our data set. Um, if we can imagine someone's eating a big salad of this almost every single day for 30 years, and we want to know what is the health risk from that? What's the health risk of growing and eating your own food over the course of your lifetime? One of the ways that we can understand and estimate this risk is using a quantitative risk assessment. So once we know which contaminants of concern once we know the contaminants of concern in the community, here we're focusing on inorganic contaminants like arsenic, lead, manganese, nickel, and chromium, and so on. Once we know what contaminants there are, we can calculate the dose. I'm not going to go into all the details of this equation right now, but this is one way to understand risk. Rooted in a settler colonial worldview that operates on a lot of generalizations and assumptions and uncertainties, but used in combinations with other ways of knowing and other ways of understanding risk and the environment can be really helpful to inform our relationship to soil and plants. So once we know what contaminants we are concerned about, we can calculate the dose, which is how much of a certain contaminant that we would take into our body over the course of our lifetime. And then once we've identified the dose, we can relate that to a response or a health response, like a cancer or other disease, using established equations that help us then characterize the risk. To explain the risk a little bit more, there are a few different metrics that we use to contextualize that. Um, these equations are showing non-cancer health risks and how we can calculate and understand them. I'm not going to focus on them right now. This presentation is going to mostly be about the cancer risks. So let's pause on that. And I'll talk about the increased excess lifetime cancer risk value. So to get this value, you multiply the dose of one or more contaminants by the contaminant's cancer slope factor, which is the dose response curve that I had mentioned earlier. That value then gives you the increased lifetime cancer risk that you would have from ingesting those plants or soil. According to the National Cancer Institute, nearly 1 out of 3 men and women in this country will develop some kind of cancer during their lifetime. And these values that I'm going to be showing you today are in addition to that, on top of that. And the government has set certain target risks where any additional cancer should be below some of these values here, or within this range or below. So 10 to the 6, you'll see these values pop up at a couple points during the presentation, means a 1 in a million increased cancer risk, 1 in a million increased chance. 10 to the minus 5 is 1 in 100,000, and 10 to the minus 4 is 1 in 10,000. To calculate all these health risks, we ran a Bayesian Monte Carlo Markov chain simulation with all this data. And again, if folks have specific questions about the data, I can go into it, but I just wanted to show you that a lot of information went into it. All the concentrations, ingestion rates, cancer slope factors, all were plugged into a software called Oracle Crystal Ball to run the analysis where we did 10,000 simulations per analyte per exposure route. So each plant had its own set of 10,000 simulations for every single analyte or contaminant that we looked at. Then all that data was plugged in and analyzed further in R. And we have a better understanding of the risk now. And I left one question mark on the figure because we still don't know everything. These are simulations, they're scenarios. It's not, and they're operating from a single methodology and framework. There are many different ways to understand risk, and this is just one of them. There's still more to look at. So let's get into some of the results. This heat map shows the median cancer risks for all the exposure routes and all the analytes. The exposure routes are listed on the y-axis. So each plant that we looked at plus garden soil is here. And the contaminants that we looked at in each of those plants are on the x-axis. For the squares, the rectangles on the heat map that are more yellow or brighter color, that means the risk was higher from that specific exposure route and analyte combination. So collard greens, cucumbers, kale, radish, and turnips had the highest risks associated with them coming from arsenic specifically. But when we add every single one of these when we add every single one of these rectangles up together, the cumulative risk, if you're eating this whole salad almost every day for 30 years, your increased risk is going to be 6.53 times 10 to the minus 4, which is 653 times higher than the target risk of 10 to the minus 6. And to this cumulative median risk, 16.5% of that is coming from collard greens alone. And you might be asking, okay, what can we do about that? It seems like we're getting a really high risk from eating plants grown in our gardens, in environmental justice communities. But now that we know what the risks are, we can rework the same calculation to estimate the concentrations of the riskiest contaminants that we should aim for in our plants and soils. So we can calculate screening levels that will let us figure out if a concentration of arsenic in our soil is below a certain screening level. That means our health risk from eating plants grown in that soil will be below a target risk. So we can rework the equation to look at concentration. And um, I want to emphasize that or highlight that when we did this calculation, we chose not to do a screening level for each individual plant. We wanted to focus on plant category, which is loosely based on morphology used by international or the World Health Organization and EPA to categorize different plants. Some of those categories are brassica vegetables, bulb vegetables like onions, fruiting vegetables like squash, fruits like berries, and so on. And plant category in most of our modeling beat out plant family as a more significant driver of contamination. They're both collinear, but plant family like Lamiaceae, where mints are, or Asteraceae, where where sunflowers are, wasn't as significant as plant category. So morphology, what part of the plant is being consumed. This figure shows the estimated screening levels compared to existing screening levels that exist. And I'll highlight arsenic and nickel specifically. So in blue, the blue dots show the estimated risk-based screening level that we calculated based on our data set for each of the plant categories. So this is saying that if your soil has an arsenic concentration on the y-axis, has an arsenic concentration of less than around 0.5 milligrams per kilogram, then your risk from eating onions grown in that soil will be less than 10 to the minus 6. And then similarly for herbs, if your concentration of arsenic in your soil is uh, around or less than 2 milligrams per kilogram, then your risk from eating herbs grown in that soil will be around or less 10 to the minus 6. And we can see that the ex calculated screening levels, the risk-based screening levels, in general are above, less strict than the existing strictest screening level we used from California EPA of 0.11 milligrams per kilogram. But the US EPA screening level for arsenic in soil, cancer screening level, is 0.68 milligrams per kilogram. And we see that it's not actually as protective of our health as the recommended risk-based screening levels from this study. So some of the existing government regulation might not be, maybe subpar to actually protect community health. And they are also not community specific. They're not using ingestion rates or concentrations of contaminants specific to the communities. And I'll quickly highlight for nickel that the existing screening levels are much higher than the calculated risk-based ones. So in this way, we have the opportunity to create place-based, community, and context-specific screening levels, as opposed to generalized ones and international regulations that currently exist that aren't only risk-based and can't actually support community health. Well, you might still be asking, I don't know how to fix my soil. What do I even do? Do I just stop gardening? Not necessarily. But we can also rework this same calculation as earlier to calculate the recommended ingestion rate. So how much of a certain plant should I eat to have a risk at our target level or below? This bar plot shows those risk-based ingestion rates. And again, I'll highlight arsenic and nickel cancer risks. This plot shows on the x-axis, again, the plant categories that we looked at, and on the y-axis, the maximum ingestion rate that's recommended from this study in grams per day. The simulation is for an adult weighing around 80 kilograms. And the results show that the recommended ingestion rates for your risk to be less than 10 to the minus, your cancer risk to be less than 10 to the minus 6 are pretty low. So if we look at this first column here for arsenic, the brassica vegetables recommended risk-based ingestion rate is less than 1.5 grams. So that's less than 1.5 grams of brassica vegetables that you can eat every single day to have an arsenic risk below 10 to the minus 6. For context, 1.5 grams of brassicas could be anywhere from 1 to 3 teaspoons. So it's not very much. And here we're even seeing herbs is even less than that. Half a gram of herbs a day is half a teaspoon or less maximum. When we look at nickel, it's similar. The values are higher. The risk from nickel is less than the risk from arsenic, but still the recommended ingestion rate for fruiting vegetables like squash and tomatoes, for example, is pretty low. Like a small zucchini that you might eat in one day could be around 100 grams, so much higher than the recommended value. So recommendations from this study are unrealistic, but they illustrate and confirm that pollution is everywhere. We can't avoid it. We can reduce our risks, but we can't necessarily eliminate our harm. Again, unless we're addressing the systems that created the harm in the first place and perpetuate pollution and extraction. But it's not as simple as everything that we're growing in environmental justice communities is harmful. When we compare the data from our study to a federal database, the Total Diet Study, of contamination and nutrition of conventionally grown grocery store produce. So when we compare that database to our own plant samples grown in environmental justice communities, we don't see a ton of differences. This violin plot is showing the distribution of all those 10,000 simulations I talked about earlier for every single analyte and for all the exposure routes or vegetables that we compared. I'll focus in on arsenic so that you can get a better look at the plot. In blue, again, is the community data, and in yellow here is the federal database, the total diet study. And the x-axis this time is showing the risk. And you can see that there's a lot of overlap between the yellow plots and the blue plots, meaning that there isn't a ton of difference between the conventionally grown produce risk and the risk from eating crops grown in environmental justice communities. And when we look at the average concentrations in that database compared to the community data, we see that yes, the average concentrations of contaminants are generally higher. The blue dots are a little bit higher than the yellow dots. But we also see that the concentration of nutrients in the homegrown produce is higher than the conventionally grown produce. So this is indicating that it's not as simple as saying that if I live in a community impacted by pollution, I shouldn't be gardening. It's not that simple. There could even be health benefits to growing and eating your own food, in addition to the other benefits of gardening and rooting into cultural practices and a relationship with land. So again, pollution is everywhere, even in food that we're eating in the grocery store. It's the result of capitalist and colonial land relations, but it is possible to do something about this. As individuals, we can make sure we're washing our produce really well, so we're not eating extra soil by accident, mopping and cleaning properly, covering up soil that we think is polluted. But our research also shows that this individual action isn't enough, and that we need to root into collective action and again change the systems and the cultures that lead to pollution in the first place. So that's what objective three is about. And I'm going to be talking about how we explored how culturally relevant environmental health and justice education can incite culture shifts and systems change to address pollution. This is where I'm going to be talking about our trainings in across across multiple communities in rural mining towns in Arizona. Our youth trainings, in addition to a host of other events and activities like surveys, listening sessions, science nights, and so on. Um, and I we're gonna talk about how these efforts can lead towards a regenerative economy, a regenerative relationship with land, in rooted in healing and and sovereignty and the ability for communities to choose their own risks and choose their own path forward, not under the control of corporations and the state. Just a reminder about all the data that's going into it. And right now, I'm going to be focusing on the column on the right, all the data from the youth trainings. So during this training, again, we had environmental health literacy activities, we had field trips, we were analyzing our own analyzing our own soil samples, doing data viz, and had guest speakers. We had lunch every single day, and it was a really, really sweet time. This is my favorite section. I'm really excited to talk to everybody about this. One of the major takeaways from this work is that rural youth living in environmental justice communities already know what's up. They already know what's going on. There is a high baseline knowledge and awareness of environmental health issues. During for our photo voice project, students submitted photos and captions about environmental health topics in their neighborhood that they wanted to, and students were bringing up things about air pollution, the mines, abandoned buildings, connecting that to health, but also discussing the interconnection between humans and the environment, talking about social determinants of health, like race, the type of the town you live in, income, employment. Students were talking about forest fires, sending in memes about how freaked out and worried they are. Um, One student said highlighted the intersecting and overlapping layers of oppression and harm that their towns experience, and said, uh, because we're in such a middle zone of all these places, all the smoke just travels towards into our town. And then they said, and the thing with the mines as well, all that stuff they just put into the air just because they can, because they're making money as a company. Students have already know what's going on. Our job as educators shouldn't necessarily be to teach students as if they don't know anything or need to be brought into science and um, indoctrinated. We need to be able to support students with the learning that they want to go towards and support capacity for action. I want to show this photo of an altar that we build, and it's not all depressing. Students also have hope, and we're really excited by the training and felt motivated. The student said, "I chose for my picture to be the shrine in the classroom. I chose it because it represents all the things that I learned that make me a better person, student, and scientist. I feel extremely grateful for all the help that I had along the 5 days, and I hope to use the stuff I learned." I want to emphasize that, environmental health literacy that students had was already high, and the training also supported it. This bar plot shows the percentage of students before and after the training via the survey we gave them. Their familiarity with the term environmental justice significantly increased as a result of the trainings. This yellow bar went from 31% to 94% of people who took the survey being familiar with the term. The number of people who were able to correctly define the term also increased. The same is true for the terms and definitions for public health and environmental monitoring. Um, and students even highlighted that they might know, have a certain level of environmental literacy, but other leaders, governments, and corporations might not. One student said, a lot of other leaders, governments, or stuff like that are really ignorant when it comes to health issues. They might even ask, they're like, oh, let me see the test, and they give you the evidence, and they're like, no, that can't be right. Like our town is perfectly fine, and they shut it away until it actually kills half the population. But young people aren't ignorant about this. This box plot shows the distribution of self-rated confidence in understanding and doing different science tasks like environmental monitoring, testing soil, and so on. As a result of the training, students' self-efficacy increased. Um, and just emphasizing we have to support young people's ability to take action and to address local concerns. This bar plot is showing, again, from the pre and post survey, people's ability, their thought on their ability to make decisions about social justice issues and about the environment in their communities. And that also increased as a result of the training. But young people still named a lot of obstacles to community change. So things like a culture of silence, stubbornness, adultism, carcerality, and control of culture and discourse in communities by mining corporations, and a lack of resources, capacity, and infrastructure all came up as obstacles or issues that young people cared about. One student said, I feel like some people are a little too stubborn to see the problems. And stubborn people are probably the biggest problem because they don't want to admit that something's wrong. And they don't want to admit that some things in this town need to be fixed. This is a really important finding from the study that young people were naming culture as a major issue in their communities. Culture, again, cultures of silence, stubbornness, racism, oppression, and it emphasizes that the individual actions we take, like educating each other and recycling and cleaning our fruits, is important, but it won't solve the issues at the root. We have to be able to change cultures. So our education efforts, our organizing, and our science has to be able to support people to change culture. I want to end by saying that we exist among the intersections of identity, culture, resource extraction, pollution and nutrition, colonialism and land back, oppression and liberation, and that this research shows that industrial resource extraction, largely caused by the state through corporations and settler land relations, does harm human health. but we can and must take collective action to protect it and change things. We have to take action at multiple scales, not just at an individual scale. I'll highlight some of the actions that young people themselves named when we did a whole ecological model of health activity. These posters are focusing on issues of air pollution and burning buildings. Students brought up things like, yeah, we can educate each other, we can talk about it with our friends and family, but we can also hold protests and petitions, change policy, and ask the government to hold corporations more accountable to clean up. Students in the burning buildings group, again, talked about education, safety protocols, and regulation. But one of my favorite quotes from the whole project, they also said that I don't want to work for the government. I want the government to work for me. And I think that kind of says it all. We have to be able to take collective action to change the situations, change our context and the material conditions we live in. And the young people want it too. So let's do it. The final thing I'll say is that I'm especially grateful for all the students who were involved in the project. It's so complicated working with young people, and it's also what makes me the most excited. One of the students in Hayden Winkleman wrote this poem, and that reads, "In a world where nature cries, Earth is dying before our eyes. A wake-up call this past week showed me the truth we dare not speak. Rivers polluted, forests ablaze, Mother Earth in a silent craze. We must rise up, take a stand, protect her with a helping hand. Let's sow seeds of change and hope in the hearts of all who cope. For Earth is a precious gift. Let's mend her wounds, give her a lift. So let's unite and make amends before our home meets its bitter end. This past week taught me that it's time to act, and that's a fact." Okay. Thank you, everybody. And thanks, yeah, to all the funders, community members, students, and everyone else involved.